Tonight it's going to be a great pleasure for me to talk to you about a subject very dear to my heart, as Eduardo has said, the development of the brain. And this follows nicely, I think, on the first lecture that Rusty Gage gave us last month, Stem Cells, the Brain's Beginning. That really, I think, set the tone for the Gray Matters series. And so tonight we look at how one builds the brain, going from simplicity to complexity. So let's get to the business here and talk at the outset about levels of complexity. We can begin by noting that if we look at different regions of the brain that are specialized to deal with different activities, language, vision, olfaction, taste, different regions of the brain, we can probably identify about 100 different regions of the brain. On top of that, we can identify a riot, perhaps tenfold more different types of nerve cells, neurons, nerve cells. These would differ in terms of their shape, their morphology, with their processes, their axons and dendrites that allow them to establish connections with other cells. These would also differ by virtue of their electrical signals, the kind of signals that they send back and forth to each other. These would differ also by virtue of the neurotransmitter molecules, the molecules that the neurons make and secrete to propagate the electrical signals. So there are quite a number of these recognizably distinct nerve cells, perhaps 1,000 or so. If we then begin to look at the wiring diagram, how does the nerve cell connect to other nerve cells? How are groups of nerve cells, ensembles of nerve cells connected? Now the numbers really begin to mount up. It's widely believed, and the evidence is quite good, that every nerve cell makes about 10,000 connections onto a variety of other nerve cells, and every nerve cell receives about 10,000 inputs coming in to impinge on it. So this is really a massive level of connectivity, probably much greater than that in the laptops that I can see out here in the audience. And approaching perhaps supercomputer capability. But the summum bonum here, the highest level of complexity, comes when we recognize the number of nerve cells in the human brain. 100 billion neurons. This is really an astronomical number. It's a number that's hard to think about. It's so big. And indeed, of course, it is an estimated number. Even our best graduate students cannot be manacled to the bench for a long enough period to actually count all 100 billion of these neurons. But this is probably quite an accurate figure, and we think that this really does represent the number of neurons in the human brain. So a real range of complexity here, going from regions, functional regions of the brain, to different classes of nerve cells defined in different ways, connections among the nerve cells, and then simply the number of nerve cells alone. What about this process of simplicity through sequential assembly? Let's just take a quick walk through the development of the human nervous system, most of which, of course, as you know, occurs in the uterus during embryonic development. Remarkably, the embryonic spinal cord, we call it the neural tube, is present after only 8 days following fertilization of the egg. So one starts off with a plate of cells. This rounds up to form a tube. The lower part of the tube is the spinal cord. The front part of the neural tube becomes the brain. This is present very quickly after the initial fertilization process. And then rapidly, neurons start to be generated at really prodigious rates. Look at this. 250,000 neurons generated every minute. Every minute. This is really an astounding figure. But of course, if we're going to get to 100 billion, you can do the math, you can calculate the minutes. This is actually not an unreasonable number given where we're going to build the brain. Shortly thereafter, by about a month, a little over a month of gestation, the 5 basic regions of the brain are formed. The 2 divisions of the forebrain at the front of the brain, the midbrain, and the 2 divisions of the hindbrain. These are all organized, all patterned and in place by a little over a month of gestation. And now we see a massive process of not only neuronal generation of nerve cells, but the migration of nerve cells to the appropriate places. We'll unpack this more a little later, but it turns out that neurons are generated in one part of the nervous system, and then they have to migrate to get to other parts of the nervous system where they will actually do their business and where they will form these many connections about which we've spoken. So that process begins, and for many regions of the brain is largely complete about 2/3 of the way through the 9-month gestation period. A remarkable feature of the development of the brain at that point, now that the brain in broad terms is largely complete, is that it needs to expand the area of this cortex, this very thin area on the surface of our brains where a lot of our computation goes on. And so it does this by folding into gyri and sulci. The gyri, the upward folds, the sulci, the little trenches in between the regions of cortex. And that occurs during the 6th and 7th months. And then finally, shortly after birth, during the first year, you may have seen this perhaps with children or grandchildren, the size of the brain doubles during this time. Not so much because of the addition of new neurons, but because of the addition of glial cells, support cells that are born after neurons and that are very important for the brain's activity. So there's a sequential process here. Implicit here is the initial induction of the nervous system at a very early time at the top, the proliferation of cells, migration of neurons, and then differentiation of cells and connectivity at these later points. So what are the 5 easy steps? What is coming? Let's just quickly walk through them and then come back and look at them each in a little more detail. The first process that has to happen is induction. Some sort of set of instructions have to be given to the nerve cells to go out and be this kind of a nerve cell, be that kind of a nerve cell in the cerebellum, in the cerebral cortex, in the basal ganglia. This induction process specifying different types of brain tissue is a very early event, and it's an event that's iterated then progressively as the brain further develops. Second step, as we've already seen, is proliferation, the generation of these large numbers of nerve cells. And many people have estimated that there's about 10 times as many of these glial cells. That would make 1 trillion of these glial cells. That may be a bit on the high side, but certainly there's an excess of these glial support cells over the numbers of nerve cells. Proliferation is followed by the process of migration that we've already touched on and we'll come back to. And then we see differentiation of the neurons and the development of these different types of nerve cells. Again, different kinds of excitability, different kinds of morphology, shape, different kinds of neurotransmitters. This is the kind of thing that's going on as these neurons are differentiating. And finally, what many people regard as the acme of neuronal development, the actual formation of connections, the wiring together of the nervous system so that the computer, if one likes that metaphor, can actually boot up and start to work and generate behavior, generate creative activity, many of the things that we enjoy doing with our brains. So these connections are really a very important step in the process of brain development. Well, let's have a closer look then at these 5 easy steps for development. Let's start with induction. This is a process that has 2 components to it. The first component of induction is brought to the developing nervous system by lineage. The progeny of particular cells contain within them intrinsic cues that contain part of the programming for the neurons. The second part of induction involves tissue-tissue interactions, 2 tissues coming together, in some way communicating with one another, perhaps through cell adhesion molecules in some cases, through growth factors in other cases. These are extrinsic cues that allow one tissue to shape the fate, the plan of instruction for the other tissue. The most dramatic example of induction, with which some of you may be familiar because it's so well known, comes from work in the last century from Hans Spemann and his student Hilda Mangold, looking at induction in the frog embryo, the amphibian embryo. And they did a very simple experiment, but it's had an enormous impact on our thinking about how the brain develops. What they did was to take an early stage frog embryo, shown here at the top left, and they took out this little piece of tissue from a very particular region of the early embryo, and they implanted it into, here in the interior, it's a cutaway section here, of a second embryo. Then they allowed development to proceed, and the remarkable finding was that in this second embryo into which this piece of tissue had been implanted, an entire second body axis developed. So here's the neural tube that become the spinal cord running along the embryo. Here are the muscle cells, the trunk muscle cells allow the larva to swim on either side. This is the primitive backbone or notochord in the embryo. If we step back and look at the output here, we see a kind of a Janus-faced, double-headed embryo zippered together along the ventral side, along the tummy. This is the primary embryo that would develop normally from this early stage, and this is the secondary embryo that's induced by the implantation of this tissue. Important to point out that this second body axis does not develop from this tissue. In fact, this tissue changes the fate of these other tissues and makes them become this second body axis. This was a profound observation, led to the Nobel Prize for the work, but it gave us a very clear picture of where we had to go and what we had to do. Now we have to find out what it is about this region referred to as the organizer region, a little like a union organizer. It's out there getting the troops in gear. This organizer region clearly has special features. Other regions of the embryo are not effective when transplanted in this way. So this is a marvelous example of induction in which instructions are given to change the fate, in this case, but under normal conditions, of course, normally program the fate of the development of the embryo. Let's turn to a second aspect of building the brain, and that's the proliferation of cells. And here, if we look at a kind of time-lapse cartoon from left to right here, we see part of the neural tube. The neural tube would actually go down to the floor here, and so we're looking just at a part of it, but we can distinguish the ventricular surface here. The interior of the neural tube contains fluid in the ventricle. We can also distinguish the pial surface, the connective tissue on the surface of the neural tube. And in the developing neural tube, cells typically extend processes that go the full width across the dimensions of the neural tube. And what we notice as we do this little time-lapse walk from left to right here is that the cell body with the nucleus of these cells moves from the ventricular part of the neural tube up to the pial region of the neural tube up here, and then it moves back down again. And after it does this a few times, it gets some signals, the identity of which we're still trying to determine, and that leads to division of the cells, producing, on the one hand, cells that keep doing this migration and getting important information, and then other cells that will differentiate into neurons. And if some of you were here for Rusty Gage's lecture last month, this will be very reminiscent of the behavior of stem cells, cells that renew themselves on the one hand, and yet can spin off progeny that will differentiate on the other hand. So this process of proliferation is what is going on at that prodigious rate that we talked about earlier. Turning then to migration, the movement of cells to distinct regions of the brain, here we can distinguish several different kinds of migration. And the first of these is lateral migration of cells in the nervous system. Migration along the surface, from the front to the back, from the back to the front, from one side to the other side. And an example of this is afforded here by this nice image from a chicken embryo. And what's been done in this case is to inject some tracer molecules, some fluorescent dye molecules into the brain and then watch where they go. So let's get oriented first. Here at the top is the brain of the chick. This will be the developing eye here after a few more days. The trunk region would extend down through the floor, and this region here is the site from which blood vessels leave the embryo and run to the yolk, and from the yolk back to the embryo to supply the chick embryo with its nutrients. What we see if we look at the orange tracer that was injected right at the tip of the yellow arrow here is that over a period of about a day after the injection of the tracer, cells that were labeled in this region have now migrated, in this case ventrally, more toward the ventral surface, the tummy surface of the neural tube, but also anteriorly. Perhaps some of you, particularly in the front, can see some orange cells that are moving up here toward the anterior region of the chick brain. In contrast, when another indicator is injected into this region of the brain, more rostrally, more anteriorly, again right at the tip of the arrow, now we see a fairly symmetrical migration of cells in all directions, radially, around the point of the arrow. So the thing that we can take home from this kind of an experiment is that different cells in different regions of the brain are going to undergo different migratory pathways, going to migrate differently to wind up at the correct sites. So this is lateral migration, a very important migration in the brain. But then in addition, there's a vertical migration that's quite important. And so once again, let's look at a kind of time-lapse series of cartoons for the development of the mouse brain, in this case mouse cortex, looking first at embryonic day 8.5, embryonic day 10, embryonic day 12, and then the adult. Remember, this is a 21-day, 3-week gestation period for the mouse. And at the outset, at the early time, we have an image that looks a bit like a slide I showed you a few minutes ago. Here's the neural tube, a section through it. Cells are dividing, proliferating here close to the ventricular margin at the base of the neural tube in the ventricular zone, VZ ventricular zone. By a day and a half later, now the structures have become a little more complicated. Some of these cells that have divided have moved up to the preplate region, PP, preplate region. The intermediate zone here separates these two regions, the preplate and the ventricular zone. 2 days later, E12, now more neurons are generated here in the ventricular zone. And many of them are migrating up along these blue fibers, which are a particular class of glial cell, the Bergman glial cell. And they kind of climb up the glial cell, migrating along it to get up to the top of the neural plate, which, as you can see, is now expanding in its thickness as a result of the migration of these cells upward toward the top. So they migrate past the intermediate zone, the subplate, the cortical plate, up to the molecular zone. And by the time we get to the adult, this process has been iterated many times, and the cells that have proliferated here at the base of the neural plate have migrated up, and the last born cells have to migrate the furthest because they migrate right on by everybody else. So layer 6 is formed first, layer 1 is formed last by the late arrivals that have migrated past layers 6, 5, 4, 3, 2 to get to layer 1. So this is a vertical migration, very important migration. It establishes the important structure of our cortex, which is a 6-layered structure in our brains, just as it is in the mouse brain. And the mouse brain has been a very useful tool with which to study this process in great detail. Well, this brings us to the fourth of our 5 easy steps, the differentiation of neurons, the development of these many different types of nerve cells. Here, one of the key features of early embryonic development in the nervous system has been the appearance of different forms of electrical excitability, the ability to generate and to propagate electrical signals. I'll come back to this in more detail later on, but as a broad introduction here, an interesting feature of many embryonic neurons has been that at the early stage of development, their electrical signals, action potentials that are the currency of information exchange in the nervous system, have a rather unusual appearance. They're relatively long in duration. This is a time axis here. Voltage of the membrane of the neuron is depolarized and then repolarizes, and one has this kind of boxcar-like appearance for the electrical signal, reminiscent in many ways of the action potential that one sees in heart muscle. We remember that our hearts generate action potentials. These action potentials are important for the regular contraction of our heart muscle. We want that to continue in perpetuity. And those action potentials very much resemble these in young neurons, long in duration, and a result of the constellation of active protein channels in the nerve cell membrane. So at the early time, there are channels that allow the influx of sodium ions, calcium ions flowing into the cell. That leads to the depolarization of the voltage across the membrane of the neuron. This is then sustained during the plateau of the action potential by continued influx through the calcium selective ion channels. And finally, the action potential is terminated, brought to a close by a flow of positively charged ions, these potassium ions, K+ ions, potassium ions, out of the nerve cell. So it winds up that these action potentials in young neurons deliver a real a bolus of calcium to the interior of the cell. It's enough to make one wonder, what could be the function of that calcium signal at these very early times? And we'll come back to that. Within a short period, typically a day or so, the action potential changes dramatically in its shape. It goes from a long event to a very brief, short event, about 1/1000th of a second in duration. Here it would be about 1/10th of a second in duration at the in young neurons. And this comes about because of changes in the expression of these protein channels in the nerve cell membrane. What you can appreciate from the cartoon is that there are now more of these sodium selective channels, allowing sodium to flow into the cells, and most significantly, a large increase in the number of potassium channels, protein channels that allow potassium to flow out of the nerve cell. So a dramatic change in the excitability of the neurons during this very early period. And different neurons display different aspects of this kind of change in excitability. This is perhaps the easiest one to appreciate. In addition, different neurons begin to express different types of neurotransmitters. This has turned out to be a cottage industry. When I was a student, we thought that there were maybe 5 or 6 neurotransmitters. Is that fair, Eduardo? The number was small. And now it's well over 50 and probably closer to 100, or I don't know, Julie, maybe 150. It's particularly when we factor in the enteric nervous system. So it's turning out to be a very interesting question to ask: how do neurons, how do they know? How is their transmitter specified? How do they make this transmitter and not any one of the other 149 transmitters? And we'll come back to that question a little later on this evening. So differentiation, development of these different types of nerve cells, very important process. And this brings us to the last of these processes, the formation of connections among neurons. How does this occur? Well, here we learned, and again, Rusty previewed this briefly in his talk in January, that there's a very important structure, the growth cone, at the tips of extending neurons. So the nerve cell has, of course, a cell body with the nucleus where the chromosomes and the DNA are, and where proteins are made for the most part. Then it has a long process, the axon, that goes out and makes connections with other nerve cells. And at the tip of the growing axon is this structure, the growth cone, an example of which is illustrated here. So the cell body would probably be over here on the wall, on the far side of the room. The axon would come cruising in here and terminate in the growth cone, roughly conical structure here. And you'll notice right away that it has a number of these fine thread-like structures that are sticking out of it. These are filopodia. They're kind of antennae, if you will, for the growth cone. They have both a sensory function to detect what's out there in the environment, and also a motor function helping the growth cone move along toward its targets. This growth cone is growing in a dish. In the laboratory, it's from the local sea hare, a plesia that you can find down here on the beach at low tides. And so this is a growth cone from the nervous system of this sea hare. Pathfinding by the growth cone then proceeds through recognition of targets along the route to the final target that will be innervated, that will be connected to by the growing axon. And here we've learned to recognize what we call guidepost cells, an example of which are shown here. These are intermediate targets that the growth cone will recognize and must have to find its way to the ultimate target. So in the grasshopper embryo, in the limbs of the grasshopper, out at the tip, sensory neurons arise very early in development. And the beauty of this system is that you can see into the limb. It's transparent. We used to do this in my lab, and you could actually identify individual neurons as they're starting to grow and see them in the microscope. What we saw was that these axons make contact with some of these guidepost cells. Here's FE1, here's TR1, here's CX1. There are two of those. These guidepost cells have a very important function. If one comes in with a laser and destroys FE1, for example, the growth cone, when it starts back here and starting to come along in this direction, gets confused, gets lost, starts wandering around out here in the limb, has a great deal of difficulty pathfinding and ultimately getting into the central nervous system where it normally arrives and makes its connection. So these guidepost cells, which have also been referred to as stepping stone cells, a nice metaphor, these guideposts or stepping stones, very important part of the process of making connections in the embryonic nervous system. And we think that this is not a peculiarity of the grasshopper, much less the sea hare. These fundamental properties that one identifies initially in these model systems turn out also to be operating in mammalian systems and where we can look at it in the human nervous system as well. Well, we're still not at the point of having made connections, but I thought I would illustrate for you some of the beautiful work of Anne-Marie Craig at Washington University in St. Louis, who is looking here at a neuron from the cerebral cortex that is growing in culture in a dish. This is very useful, of course, for a number of experimental purposes. And in this young neuron, at an early stage, other nerve cells that are out of the field of view here have made only a very few synapses. And these synapses, these synaptic connections, are indicated by the little green spots. There's an antibody to a molecule that's localized at the synapse. The antibody can be tagged with a nice green indicator so we can visualize the individual synapses on these dendrites, these thin processes of the neuron. If we look at another culture some weeks later, now the neuron is much more mature, and now we see many synapses. I'm not sure that there are 10,000 here yet, but of course this is a somewhat artificial situation in the dish. But we certainly have many more of these little green spots on the dendrites of the nerve cells. And some of you may be able to notice also that there are some interesting red spots up here on this very faintly stained dendrite. These represent a different class of synaptic inputs that are recognized by a different antibody to a different molecule that is tagged with a red fluorophore, a red dye, so we can see that particular population of the synaptic inputs. So over time, the number of synapses that are formed on the neurons increases, and we get a very large number shown here. Well, it's been interesting to learn, and actually this is a fairly recent finding in the past few decades, that not only are these synapses important for rapid communication back and forth, the give and take of human interactions, but they're also important for survival of the neurons. If nerve cells don't make synaptic connections at the early stages, typically they don't do well and often they die. So much as we in society, in human society, develop great sustenance as social beings from our interactions with friends and colleagues, family members, so also neurons need to be connected in order to survive. Well, that's the 5 easy steps, and again, I think that it's possible to fold a lot of complexity into this sequence of events that we see, but I want to move on now to the observation has been that cell adhesion, the sticking together of cells via particular molecular interactions with molecules that Jerry and his colleagues have been able to identify, in turn regulates their structure, the structure of the neurons, and that, by means that we don't yet understand, governs gene expression. So one goes from interactions to morphology to gene expression. And this has turned out to be a very interesting process. In some sense, there's a mechanochemistry, the mechanics of the chemistry of interaction of molecules that's linked to the developmental genetics, the turning on of different genes. during development. So how might this work? Well, one would imagine that cell structure is going to drive gene expression. One would start with induction, the kind of thing we've looked at together. That in turn would lead to the expression of cell adhesion molecules, these cell adhesion molecules, CAMs. Those in turn would then lead to changes in the morphology of the cells expressing these cell adhesion molecules, because they're now going to stick to some cells and not to others and get pulled and stretched in different directions. That in turn would lead to regulation of gene expression. This is the least well understood link in the process, but that in turn changes the molecular context of the cells so that the process of induction can occur again. And one can go round and around this cycle many times, and certainly does so during the development of the brain. A second kind of dynamic process that I want to describe for you this evening involves transient elevations of what are called second messenger. In biology these days, as some of you will be familiar, we think of signaling proteins, small molecules that come in the blood, hormones, neurotransmitters. We think of these as the first messengers. But it's a little like running a relay race. If one wants to carry information into the cell interior to change things inside the cells, we have to pass off the baton to someone else, a second messenger, who can then carry the information further into the cell interior. And it turns out that transient, brief elevations of second messengers, such as calcium ions or cyclic nucleotides or a variety of different components, regulate enzyme activities that can either determine cell structure or, in other cases, govern gene expression. So here, the brief message would be that these second messenger signals can regulate neuronal differentiation either through cell structure or gene expression. And again, the dynamic would look something like this. We would start with induction. This would then lead to the appearance of some form of electrical activity. This would lead then to elevation of, let's say in this case, calcium ions inside the cell for a brief period of time. That in turn would lead to regulation of the cytoskeleton, the skeleton that's inside the cells and that governs their morphology, and to gene expression. And once again, by changing the molecular context of the neurons, this would then feed back to induction, and again, one can go around this loop a number of times during the assembly of the brain. So in the last part of the talk now, I'd like to focus on the theme that has been very dear to my heart and illustrates some of these principles and discuss with you a little bit about what we know about the roles of electrical activity in building the brain. The first piece of work that I'd like to tell you about focuses on the way in which the outgrowth of the axon and the migration of those growth cones that we were talking about earlier, how that is implemented by transient elevations of calcium that occur and are localized in the growth cone itself. Here's what the embryonic nervous system of the frog looks like, seen from the side after suitable staining to visualize different structures. At the bottom here, indicated in green, is that structure we talked about earlier, the notochord, the primitive backbone. It contains a lot of the protein actin, which is, of course, one of the two components in our muscles. Actin and myosin are what make our muscles contract. Here, there's no myosin, just the actin, and it's revealed with an antibody that has a green tag on it, so we see green actin here in the notochord. Above the notochord is the neural tube, and shown in red here at the top, on the dorsal surface at the top of the neural tube, are cell bodies of neurons that are stained for tubulin. This is one of the major girders, sort of the I-beams of the construction of the neuron. We find that in the cell body. We also find it in the axons that are running along the dorsal surface of the neural tube. And of course, as many of you will recognize, where actin in green and tubulin in red come together, red plus green makes yellow. And so we see a lot of yellow here in the intermediate part of the nervous system. And I'd like to draw attention to two interstate highways. This bundle, this fascicle at the top here, maybe that's 805, and then this other one down below, maybe this is I5, or maybe I15, I'm not sure. But in either case, these are major bundles, lots of axons flowing down these tracts. And here, coming down across these tracts would be route 52, route 56. These are axons that are coming down around the side of the neural tube to cross over and make connections on the other side. So this is kind of an overview of what the nervous system really looks like. This is a cartoon now to try to capture elements of that picture. We can see the dorsolateral fascicle, the ventrolateral fascicle, those interstate highways, 805, I5. And then we can see different classes of neurons. There are only 8 classes of neurons in this embryonic nervous system. That's one of the reasons it's a very attractive system to study. There are so few neurons, so few classes. We can see these sensory neurons, the ones that were stained in red with tubulin in the previous slide. Here they are. Their axons run in the dorsal fascicle. I would draw your attention particularly to the motor neurons, here labeled MN for motor neurons. They send their axons in the ventrolateral fascicle. And then I want to draw attention to this final population here, the blue population. These are dorsolateral anterior neurons. They are not on the freeway. You'll notice here that the freeways are up and down, but they're on the back roads here driving around in the country, and that'll be important for what we'll be showing in the next slide. But once again, here's a real nervous system, just so you can see that this is not a total tissue of fantasy. Here's the sensory RB neuron at the top. This is the dorsal fascicle, which forms early. The ventral fascicle is just forming here. You can see a bit of it at the left. It hasn't formed in this other region. Here's a motor neuron cell body corresponding to the motor neuron here, sending its axon down onto the freeway. This is the on-ramp, one imagines. And then here's the dorsolateral ascending interneuron here, the DLA, with its axon running in the interior of the neural tube. So let's look at a couple of movies so that we can see what's going on here in the growth cones of motor neurons and in the growth cones of these DLA, dorsolateral ascending interneurons. The procedure, very simply, is to load the cell bodies here by creating a little cavity in the embryo, loading the cell bodies with a calcium indicator that reports in its fluorescence the level of calcium in the neuron. And then we capture these movies and pseudo-color them so that purple and dark red are low calcium, and when it turns bright red, that represents an elevation of calcium in the growth cone, which will be indicated by these little arrows here. So let's start the movies, and let me just advertise what I think you'll see. You'll see that the motor neuron growth cones grow really rapidly, and they don't make these growth cone calcium transients, whereas the dorsolateral ascending neurons, they generate these calcium transients out in their growth cones. So this is the kind of imaging that we get. Watch right here. You'll see that there's a flash. There's another. And there'll be more here from this growth. There's several more. Whereas in contrast, the motor neurons here, which are going like bandits, don't generate these growth cone calcium transients out in their periphery. So from these kinds of data, we learn that the activation of the growth cone, calcium flowing into the growth cone through calcium selective channels, elevating calcium just locally in this region, not spreading to the rest of the nerve cell, governs the rate of outgrowth of the axons. And so here's a very simple graph in which we look at the rate of outgrowth, microns per hour. There are 1,000 microns in a millimeter, so this is pretty slow growing here. 50 microns per hour would be 1/20 of a millimeter per hour. But we see that it's inversely proportional to the frequency of these transients. Low frequency of calcium transients, as for the motor neurons here, leads to a rapid rate of outgrowth. A high frequency of these growth cone calcium transients, 10 to 15 per hour, as for the DLA interneurons, leads to a relatively slow rate of outgrowth, perhaps 5 or 10 microns per hour. So this early form of excitability, localized to the growth cone as its pathfinding, looking for guidepost cells. This kind of excitability acts as a set of brakes to kind of damp down the rate of outgrowth so that the growth cone can check around, sniff here, there, palpate the environment, find out where it needs to go, recognize the stepping stones, and move on in the right direction. A second class of calcium transients that we've worked on is again localized in the growth cone, and it involves the turning behavior as the growth cone turns toward or away from an intermediate or final target. And these are located out in the very fine filopodia that we drew attention to earlier. Here's an example of what one can see in culture when one takes nerve cells from the frog embryo, puts them out in a dish. The cell body for the neuron would be over here on the right side of the auditorium. The axon would run in here, run in from the cell body. And then on the dish, it's coming up to an artificial boundary that we've made in the dish with laminin, a biologically relevant molecule found in the brain. On the right-hand side, it's been labeled with rhodamine, a red fluorescent indicator, so we can see where the laminin is located. And on the left side is simple tissue culture plastic. This is commercially available plastic substrate. And what we can see is that the axon comes in as if it were going to cross the boundary, and it turns. It doesn't like tissue culture plastic. Perhaps no surprise, neither do we. But it turns to remain on the laminin. It stays on the laminin. Those of you in the front may be able to see that there are little fine filopodia that are extending across. The growth cone is trying to palpate what's going on over here, and it's clear that it doesn't like what it sees or feels or in some way senses. So we've looked into this to try to understand how this guided behavior is mediated. And this is another movie that will show us the activity in the very fine filopodia of the growth cone. So again, one has to imagine that the cell body for the neuron is way up here at the top of the roof of the auditorium this evening. Axon comes down here and then runs out toward us, coming toward us in the audience. Here, calcium is coded in two ways. Part of it is coded by color. So red-white means high calcium. Blue-purple means low calcium. This is a pseudo-coloring process. But the calcium levels are also coded by the height of the plot here. So this looks a bit like the polar ice cap here in white at the middle, that high calcium level there in the growth cone proper. Here in the fine filopodia that are extending out from the growth cone, calcium is typically lower. So we're going to play a quick movie here that will show us, the activity in this particular filopodium. Watch this particular one here and the one that crosses over it here. As we see here, as we watch for a little while, we see that there are elevations of calcium, evident both by the color and by the height of the profile, that are initiated often out here at the tip, and then propagated back to the growth cone proper. There goes another one. So there are evidently signals that are triggered out here in the periphery in these filopodia and then propagate back to the growth cone, in some cases stimulating elevation of the larger growth cone transient that we looked at in the previous movie. So it was of interest then to find that the frequency of these filopodial calcium transients that we just saw in the movie, the frequency depends upon the different substrates on which the neurons are grown. So in that earlier slide, we compared the behavior of the growth cone on laminin, which the growth cone liked, with the behavior of the growth cone on tissue culture plastic, TC here, tissue culture plastic, which the growth cone clearly was trying to avoid. And what we see is that the frequency of the filopodial transients here on tissue culture plastic is higher than it is on laminin, but we get a range of different frequencies for these different cell adhesion molecules, L1, cadherin, and laminin, these extracellular matrix molecules, fibronectin, vitronectin, and tenacin. And for tenacin, which is clearly the most effective agent in terms of increasing the frequency of filopodial transients, we can see that as we increase the concentration from 5 to 10 to 50 micrograms, just a measure of the amount of the tenacin on the dish, the frequency of filopodial transients increases. So now we're in a position to actually do an interesting experiment here together this evening, and that's to look at how the growth cone behaves dynamically when we force it to approach a realistic boundary between two different concentrations of the same molecule. And that's shown here. Again, we've established condition in a dish. The neuron cell body would once again be way off the screen over here to one side. Axon coming in. Here's the growth cone labeled with the calcium indicator. And for our viewing pleasure, 3 of the filopodia are indicated here, A, B, C, and boxed at their terminus, which is going to be the region that we're going to be imaging filopodial transients. In black, this region of the dish contains 5 micrograms per milliliter of tenacin, low concentration. At the bottom, indicated in red, a much higher, 10-fold higher concentration of this biologically relevant molecule that we find in all of our brains. And what we can see is that sampling the frequency of the filopodial transients under these different conditions, under the conditions indicated by A and B, the low concentration of tenacin, low frequency of filopodial transients. Over this 25-second period, just a handful. In contrast, on the more concentrated substrate here, 50 micrograms per milliliter of tenacin, a substantially higher frequency of these events. And when we look under the control condition where these transients are generated, 79% of the neurons come down to the boundary, don't like it, and turn away. But if we block these calcium transients with an agent that acts like a sponge, it just soaks up the calcium, the filopodial transients disappear. Under those conditions, now only a third of the growth cones will turn away, and 2/3 of them come down to the boundary and happily cross right on over and keep going on the higher concentration of tenacin. So this kind of an experiment, manipulating the calcium transients in these growth cones and looking at the consequent turning behavior of the growth cone, established, we think, fairly firmly a role for this calcium signaling, this early form of excitability, localized just to these fine tips of the growth cone in the growth cone turning process. Well, in the last part of the talk then, I want to focus on the third and last kind of excitability under the general aegis here of our discussion of the role of activity in brain development, and talk about the specification of transmitters by these calcium spikes. These are calcium elevations that come from the long duration calcium-dependent action potentials that we looked at earlier. So here's an example of a cell in culture, neuron in culture, its cell body here, its axon here, and the growth cone at the top. And over the period of imaging here, we'll see a series of lightning flashes, bolts coming out of the sky, representing elevations of intracellular calcium that occur throughout the nerve cell because the action potential is a propagated event, unlike the growth cone transients and the filopodial transients. This is propagated throughout the entirety of the cell. And so we see these flashes of lightning, if you will, the elevations of calcium propagated throughout the neuron. Neuron's still able to grow out and extend its axon during this period. Does this happen in vivo? A remarkable finding has been that yes, the same thing happens in vivo. Here we don't see the axons yet. It's at a slightly earlier stage. But in this view, looking at the ventral surface of the neural tube, again using calcium indicator to register elevations of calcium, which show up as gold signals here on the backdrop of the blue and purple, we can see that there are a number of nerve cells, particularly at on either side of the neural tube, the spinal cord, that are spontaneously elevating their calcium and pumping it back down again, elevating it, pumping it back down again. These are calcium spikes produced by those calcium-dependent action potentials generated in these embryonic neurons. Well, to begin to do analytical science here, we want to do some experiments in which we change those firing patterns for the neurons and ask what the consequences are for their development. And what we can do is to inject at the very early stage, the 2-cell stage of the embryo, we can inject RNA that encodes different ion channels, and in this way either suppress or enhance the activity of the neurons. This is quite an effective procedure. Here's the control condition. This is like the neural tube you just saw. The active cells have been circled here with white circles to allow us to appreciate the cells that were active during a 1-hour period. When we inject messenger RNA for potassium channels to suppress calcium spikes, now we see no little white circles. Activity has been completely eliminated. If we express ion channels that make sodium channels, we see much more activity here, higher incidence of firing during the 1-hour period. So then we can go ahead and cut sections through the nervous system at a later time and ask, what are the consequences for expression of neurotransmitters following these manipulations of activity that we've done on the amphibian embryo? And what we can see is that in control conditions, in an untreated frog embryo, if we cut a cross-section through the neural tube, illustrated here by the white dashed outline, we can find cells that are positive for glutamate, an excitatory neurotransmitter, and for a marker, HNK1, again, red plus green makes yellow, and we see that's just localized here at the dorsal surface at the top of the neural tube. Similarly, if we look at another kind of transmitter, acetylcholine, the transmitter for motor neurons that innervate our muscle cells, we find that that's nicely localized here at the bottom, at the ventral surface of the neural tube, and co-localized with a nuclear marker inside the nucleus of the cell, LIM3, a transcription factor that's a marker of cell identity. Again, we don't see any acetylcholine up here in the dorsal part of the neural tube. But gee, things change if we knock out spikes. If we knock out the calcium spikes, as we saw, now all of a sudden we find glutamate down here in neurons where it shouldn't be there. And we find acetylcholine up here in the more dorsal regions of the neural tube. Again, not normal, not normally found there. And just the reverse occurs if we knock spikes in, increase spikes by overexpressing sodium channels. Now the normal expression of glutamate and acetylcholine is gone. All we have are the markers for these cells. So this has been a very interesting finding and stands up also when we look at inhibitory transmitters. Glutamate and acetylcholine excite the target cells that they innervate. Glycine is an inhibitory transmitter. It suppresses the activity of the neurons that it innervates. And in control conditions, you can see a certain number of purple cells, about 50 of them per 1/10 of a millimeter of the neural tube. If we suppress activity, that number goes down. If we increase activity, that number goes up. So there's an activity dependence both of the excitatory and of the inhibitory transmitters. We can summarize this in the following way. It turns out that these different classes of neurons generate different patterns of calcium spikes. We haven't taken the time to go into that this evening, but there are very distinct patterns of activity in the different classes of neurons. And when we change these patterns, we change transmitter expression so that when we suppress activity, as you saw, the numbers of neurons making the excitatory transmitters, glutamate and acetylcholine, goes up. And the numbers of neurons making the inhibitory transmitters goes down. Just the reverse occurs when activity is enhanced. And so this process of specification seems to be homeostatic. It seems to be trying to maintain a constant level of activity in the nervous system. So when activity is suppressed, more neurons make excitatory transmitters. When activity is enhanced, more neurons make inhibitory transmitters. Try to get back to that comfortable balance, get back to that homeostasis. And the model that we have here, this is actually not a creature from 100 fathoms that just washed up on the beach. This is our cartoon neuron with its cell body here, the nucleus, the dendrites, long axon ending in a growth cone. The model, the way we think about this process is that there are proteins that regulate gene expression in the nucleus. We call them transcription factors. They program the expression of ion channels that lead to patterned activity, which is then modulated by external signaling proteins. And depending upon whether the activity is a high pattern or a low pattern, we get different signals that go back to the nucleus. The high frequency signal indicated here in red, low frequency signal indicated here in green, these calcium spikes propagating information back to the nucleus, and then finally leading to changes in transmitters. So let's just take one example here. The high frequency of activity sends the red signal and leads to the increase in appearance of inhibitory transmitters. So this is a homeostatic model, and there's lots to be done here, but the important message that I would leave you with is that this form of electrical activity clearly has a significant role in specifying the neurotransmitters, the signaling molecules that are important for propagating information in the nervous system. So in closing, what have we done together this evening? We've looked at a series of issues related to the building of the brain, levels of complexity, perhaps at least at a conceptual sense, sequential simplicity. 5 steps, how easy? Well, I'll let you be the judge of that. Certainly a very dynamic process, and a process in which there are a number of roles for electrical activity. So several quick conclusions. For me, it's clear, I hope for you also, the brain is pretty complicated. But I hope we've been able to work through that a little bit and understand that this complexity has perhaps an underlying simplicity by virtue of serial and sequential assembly. There's a long list of components. We really touched on only a few of them this evening, but I think at the conceptual level, the steps by which the brain is built are at least relatively straightforward. This process is dynamic and involves electrical activity. In my final slide, I'd just like to make a couple of sort of philosophical comments, and this is kind of by way of a post-log after we look back here. One issue that confronts any developmental biologist, and certainly a developmental neuroscientist like myself, is nature or nurture. Which is it that's important for governing human behavior? And with my children, this is a constant struggle to try to understand the relative roles of these components. But I think the view in the community now is that almost universally both of these processes are involved. And the real question is, what is the balance between nature, gene expression, and nurture, activity, and the role of the environment on the other hand? A second point that I really do want to try to leave you with as a major take-home message is that this sequential nature of brain development allows initially expression of genes that alter the structure of the brain, and this then enables new interactions among the cells and also electrical activity, which allow expression of further genes. And we had a cartoon summary of this under the aegis of developmental dynamics. That kind of process, that kind of iterated process, very important for brain development on into adult life. And finally, again, from a philosophical point of view, I think it's probably the case that this combination of nature and nurture provides a nice way in which to allow us to make adjustments for changes in our experiences, changes in the environment that we encounter through our lives, and that this may be, at the end of the day, a very powerful evolutionary advantage. So I'll just close by acknowledging my colleagues. These are the wonderful friends doing outstanding work with me in the lab at the present time. This is a great group of people. Recent colleagues who have been with me but have now moved on to other higher ground are illustrated over here at the right. So with that, I'll stop. Thank you, and take any questions.